Hi, thanks so much for taking the time to hear a little bit about a clinical research study that is happening at Cincinnati Children's Hospital, where I am the principal investigator, and where we're talking about nutrition and metabolism in Fanconi anemia and whether or not we can sort of manipulate this metabolism to our benefit. And so, before I really get into it, I want to talk a little bit about who I am and who my lab is, and who the individuals that are really sort of the heart of the study are. And so, first off, my name is Dr. Lindsey Romick-Rosendale. In addition to working at the hospital, I spend pretty much all of my free time with the two little ladies that you see in this picture, my daughters, Lily and Annie. I have two members of my lab, two senior research associates. And the first person is Mickey Watanabe Shyland, and she spends a lot of her time with her husband Chris and with their fur children, Kai and May and Henry. And then I also have a wonderful research scientist, Sarah Vicente Munoz, who actually hails from Valencia, Spain, and who has joined our team more recently, and who will be doing really the bulk of the downstream analysis of the very complex data that will sort of come of this study. So I really want to get right into things. I don't want to take up a ton of time, but I want to talk about metabolism. And so our bodies are constantly reinventing themselves in this perpetual state of loss, but also always rebuilding. And even though all of this is happening at the cellular level, the consequences of these things happening couldn't be bigger. So the two sets of reactions that I mentioned, the building up and the breaking down, together, that's catabolic and anabolic reactions, and that's what makes up your metabolism. So, what really is metabolism? Well, people talk about metabolism in terms of how fast your body burns the fuel in your food or how high your personal energy level is. And that's fine for an article in a personal fitness magazine. But physiologically speaking, metabolism really describes every single biochemical reaction that goes on in our bodies. Now, these molecules that our body is constantly breaking up and then rebuilding only to have them break apart again are what we know as nutrients. And these nutrients are the materials the body needs to build, maintain, and repair itself. Now, there's six major groups, and while all of these are important, there are three that everyone always talks about, and that's what we see on our food labels. And these are carbohydrates, lipids, and proteins. Now, rather than go into excruciating detail and list every compound represented within these groups, we're going to focus on the important things. The main thing that you need to know is that the monosaccharide glucose is the be-all, end-all molecular fuel that your cells are going to need to make ATP. ATP being the molecule that your cells use to drive those anabolic reactions we talked about, like when you need to make new polymers or get basically anything else done, whether that's operating a sodium potassium pump or detaching the head of a myosin filament to contract a muscle. But ATP is too unstable to store. So your cells will often store energy in the form of glucose, which they then can catabolize or break down and convert to ATP when they need it. Now, we're talking about the cells of a so-called typical individual with no underlying metabolic dysfunction in this case. And in addition to getting energy from ATP, some of your cells can also get energy from fats. But many of the most important ones feed exclusively on glucose. Now, let's take your brain, for instance. Now, your brain's like a car, right? A car needs gas, oil, brake fluid, and other materials to run properly. And your brain also needs special materials to run properly, like vitamins, minerals, other essential chemicals, and glucose. The fuel or energy for your brain is glucose. You can get glucose by eating carbohydrates or other foods that can be converted to glucose. But if it's not needed right away, then that energy can also get stored as glycogen in your liver and muscles. Now, again, your body is always working to ensure that you have the nutrients needed to make things like ATP for energy. And if your body doesn't believe it already has what it needs, it's going to resort to catabolizing or breaking down sometimes important molecules to then later use the breakdown products to rebuild what it thinks it's missing. So, yeah, there's a whole lot going on within our bodies constantly that we're completely unaware of. Now, our bodies don't handle every food and nutrient that enters in the same way. After you're done digesting, some nutrients will go straight to your body's pile of stuff to burn right away. But others will have to be converted into something else entirely, right? That's how this all really works. Now, for example, the carbs and fats in your buttered toast can be directly oxidized into usable energy. But the amino acids in the bacon have to be converted into molecules to get broken down, like carbohydrates, if you want to get energy out of them. So, as I mentioned before, the molecules in your body are constantly changing shape and renewing and rearranging themselves to either build things or to use energy, and eating food replenishes these nutrients, especially glucose. Then, depending on what your body needs and what you last ate, certain hormones like insulin will help decide what to burn and what to store for later. Now, this, of course, is an important function of hormones, which is why things can go badly if the process doesn't work properly because of an underlying metabolic disorder. Now, exactly how glucose levels can spike or plummet, how we convert nutrients into energy, and how all of that relates to eating and hunger and weight, metabolism, and your overall health in general is complicated. But since we're all living things that have to eat in order to stay alive, I think that it's important that we really focus and better understand this area of work. If we're talking about a typical individual, when it comes to how this energy that we're talking about converting and where, it depends largely on when that person ate their last meal. We all switch back and forth between two nutritional states: the absorptive or fed state, which is during or after eating, and the post-absorptive or fasting state, when the GI tract is empty and the body is running off stored supplies. So let's say you just finished dinner and you're still in the absorptive stage, and your digestive system breaks up the foods into a bunch of glucose molecules that pass into the bloodstream. The first bit of the glucose gets delivered throughout the body, and it's tapped to generate ATP or energy on the spot through cellular respiration. But that's not what happens to all of it, and you've got a lot of extra glucose floating around in there than your cells really need at that moment. Now, energy in the form of ATP, as we mentioned, is too unstable to be used for storage, which means all of that extra glucose is going to have to be stored as fat or glycogen. And that storage is part of how you can end up gaining weight, because how much energy gets stored depends partly on your basal metabolic rate. Now, normally, if your body senses that glucose levels are too high, then a series of events will be triggered that will take them back down. And at this point, there's a shift from catabolic reactions to anabolic reactions. For example, it puts a stop to breaking glycogen in your liver and muscles to release glucose for energy, and instead it ramps up the process of glycogenesis, where extra glucose is linked together to form glycogen. It also activates the lipogenesis, where the liver converts glucose to triglycerides, and then it ships them off to be stored to our adipose tissue. So let's say that your body has put all of the glucose, lipids, and proteins where they need to go, and you can just coast into that post-absorptive or sort of fat burning stage. Several hours later, even though your small intestine is still working hard, your cells have been helping themselves to the remaining glucose in the blood, and eventually your blood sugar level will start to drop. And then this will trigger the opposite processes to happen, resulting in the release of glucose, fatty acids, and glycerol back into the bloodstream. Now, if it's been a few days since you've eaten for some reason, and you've used up all of your sort of glycogen stores, and you don't have any sugars left to feed things like your brain, your body will launch into what's called gluconeogenesis. And it's going to start converting fats and amino acids into glucose so you can get energy to your brain. But is this the norm? That's really the question. What if something has gone completely awry and our bodies prefer or are forced into functioning as though it is in a state of prolonged fasting, even when it isn't? What if our bodies simply continue to store glucose and break down fats, proteins, and amino acids for energy? Does my body and the body of a person with Fanconi anemia process nutrients in the same manner? Are the same metabolic processes turned on and off for both myself and a person with FA? These are the questions that we're working to answer with our research study. Before we dive into the study itself, I'd like to very briefly introduce the scientific technique that is going to largely be utilized. Now, my area of research is called Metabolomics, and while the word was made up in the late 90s and isn't the easiest to pronounce, it essentially means that I study all of those small molecules that we talk about in the first few slides, like glucose, fatty acids, amino acids, ATP, etc. Now, the great thing is that much is known about metabolism and the complex pathways that are constantly at work in our bodies. And so this allows us to create metabolic maps when persons with a metabolic dysfunction are discovered. We essentially know how the body's metabolism should work to function properly. So we start tweezing apart what's gone awry or what's gone wrong, and we work to fix that when it doesn't work properly. Now, we can try to understand the metabolism of different organisms by analyzing what is inside of a number of different biological fluids or tissues. We simply process a sample, put it inside of our nuclear magnetic resonance or NMR spectrometer, and obtain a metabolic profile of that sample. We can then go on to get quantities of compounds like glucose and lactate in a given urine, blood, or tissue sample, and then we can compare these quantities across hundreds of patients and between individuals within a healthy and a diseased population. Now, if you come to visit us, I may end up putting you to work like my good friend Jack Temperley, whom some of you may know. And this is just a little bit bigger picture of Jack standing by our 600 megahertz high field magnet, and he was kind enough to come visit us last year. Now, we'll be using a technique called or known as stable isotope resolved metabolomics. Now, this is sometimes also called isotope tracing or metabolic flux analysis, but the idea is the same, in that we can actually trace individual atoms of carbon that make up that glucose molecule that we talked about. So we can trace the labeled glucose an individual within the study ingests, all the way from glycolysis and the cytoplasm to the Krebs cycle and mitochondria, and then to lipid biosynthesis in the cytoplasm. All of these important molecules that play a role in cellular respiration and allow for our bodies to function optimally are traceable to determine their fate. So we can trace these molecules through the process of cellular respiration in persons with Fanconi anemia and without, and work to understand what's different and how we can go about fixing these things, so to speak. So the study design is actually pretty straightforward. You'll come in first thing in the morning so that you're fasted. That's very important. And we will collect baseline urine and blood samples and perform a few standard body composition assessments, like checking your grip strength with a dynamometer, measuring skin folds with calipers, and then we actually have this really cool technique where we use a machine called indirect calorimetry, and you just simply breathe into that machine normally, and that's going to actually measure your oxygen consumption and carbon dioxide production. And so it actually tells us, by measuring these things, whether a person's body prefers to use carbohydrates or fats for energy, and that's really important. So then the study participant is going to drink a concentrated sugar water drink where, instead of standard glucose, we have actually labeled the carbons of the glucose with that stable isotope I mentioned before that we can trace. And then we will also collect midpoint urine and blood samples at approximately 2 hours. And then we'll repeat the body composition assessments again, and we'll collect at an endpoint blood and urine samples as well. And then we will treat you to lunch if you are really tired of hanging out with me and the rest of my staff scientists for a few hours. So what is the scientific impact? Well, we believe that elucidating for the first time the molecular mechanisms and metabolic pathways that are impacting this nutritional status of persons with Fanconi anemia will assist in the development of novel therapies, nutritional approaches that are going to improve the overall physical and emotional health, as well as the body image of persons with Fanconi anemia. And in the future, we can then continue to use NMR spectroscopy to monitor responses to novel treatments and to better understand newly identified metabolic abnormalities. So if you attended the scientific symposium and adult meeting 2 years ago, then you were lucky or unlucky enough to see me in a wonderful spandex bodysuit where I competed in FARF tank. And this is what ultimately then led to me applying for funding and receiving wonderful funding from FARF to be able to complete this work. I can't promise you that I will put on the suit again, but it is a possibility. And so again, this is just the very sort of basic overview of what the study is. This is my contact information, my work or cell. I can be reached at either one. I would love to speak to everyone who has questions or concerns, who would just like to get a better feel for what the study entails personally. So please reach out by phone or by email, and I hope to hear from you soon. Thank you.